Thank you, Dr. Brumbach, and thank you to you and Dr. Clark and Kai for organizing this session and making the transition to virtual at the last minute. I'm really honored and excited to be here and to share with you all my work today on which we use clinical sensory phenotypes to understand the mechanisms underlying neurodevelopmental disorders like autism. And I have nothing to disclose, so we'll jump right in. The first thing I want to do is sort of give you some context for why I am so passionate about sensory disorders. And the first reason is because they are a common feature of almost every neurodevelopmental disorder that we see in our clinics. So you're probably not surprised to hear that around 80% of children with autism spectrum disorder exhibit sensory impairment, as shown in the second bar in this graph. But what might be more surprising to you is that about 80% of children with Tourette syndrome also have sensory impairments. And so we see, looking at all the different diseases shown on the x-axis of this graph, that sensory impairments are very common, even in epilepsy and ADHD, where 40 to 50% of children will have some sort of sensory impairment. So it's important that they're common, ubiquitous across the children that we see in our clinics, but are they impacting quality of life? And the answer is yes. In this 2019 study by Kojovic et al., they looked at a group of children with autism whose data are shown in red compared to typically developing children shown in green. And on your left, what you're seeing are total sensory score. So as is well established, the scores in the children with autism were lower, indicating more sensory issues than the typically developing children. And this was true across all the sub-areas of the sensory profile, which they used in this study. The authors then went on to correlate these sensory features, now shown on the y-axis, so the total score on the sensory assay, with measures of daily life, in this case, daily living skills. And what you can see is that children with worse sensory scores, lower numbers, also had more impairment in their daily living skills. Now, you might say to me, Dr. Lyons-Warren, that's just because kids who are more impaired are going to have worse performance in every area. But that's not true. In this study, when they compared sensory scores with communication scores, there was no correlation. So it's not just children that are worse off overall have worse scores in everything. It's that sensory issues specifically impact quality of life. And so from this, now that we know that sensory impairments are common and debilitating, this has led me to wonder, how do these sensory challenges manifest in specific neurodevelopmental disorders, which will be the focus of today's talk. So my objectives today are first to describe the patterns of sensory impairments in various neurodevelopmental disorders, and to give you some tools for identifying these challenges in your patients. And then at the end, I'll talk about how we use these sensory features to identify subgroups of patients for targeted treatments and to understand specific mechanisms in neurodevelopmental disorders. So starting with specific patterns of sensory impairment, this is the specific, more detailed data from the study I showed you earlier, in which they took a group of 75 children with Tourette syndrome and measured their sensory impairment using the short sensory profile. We're going to be talking about sensory surveys a lot, so I want to take a moment. On the x-axis, you'll see the specific subsections of this assay. They ask questions about tactile or touch, taste and smell, movement, under-responsiveness, auditory filtering, low energy, visual or auditory mixed impairments, and then there's a total score. And on the y-axis, we're plotting the percent of children who scored in the definitely different in the dark blue bars, probably different in the light blue bars, or typical performance. So these assays are well validated, and when you get the child's score, it tells you, depending upon the range, which of these categories the child falls into. And what you'll notice is that about 60% of these children were scored in the definitely different category for their total score, and 20% in the probably different. So 80% of children with Tourette's disorder in this study had some sort of sensory impairment. And when I first started looking into sensory processing, I thought, it's probably going to be tactile, right? Because Tourette's is a movement disorder. But in fact, what we see is that the most common area of sensory impairment in these children is an auditory filtering. So let's look at another neurodevelopmental disorder, autism spectrum disorder. and has been well documented that children with autism spectrum disorder have multiple sensory impairments. It's part of the diagnostic criteria. Dr. Sai did a great job setting me up earlier today. What was interesting about this study is they looked at low functioning versus high functioning children with autism and compared them to children with other developmental disabilities or typical development. And here we're plotting the percent of children in the study who fell into the affected category in auditory in green, visual in blue, touch in red, or smell and taste in black. And what you can see is that for low functioning autism, touch is actually the most common sensory modality impacted. But when I look at this, I still see a lot of heterogeneity. There are a lot of different sensory areas impacted across all these subgroups. So this led us to hypothesize that children with monogenic neurodevelopmental disorders would be less heterogeneous if it's coming from a genetic cause. And I'm so grateful to Dr. Holder, who did this study with me. He already introduced Phelan-McDermott syndrome to you, so I don't need to go over it in detail. But briefly, it's caused by a disruption in SHANK3, is characterized by neonatal hypotonia, characteristic hand and facial features, and frequently a comorbid diagnosis of autism. In this study, we had 41 subjects with Phelan-McDermott, and then we also included children with SYNGAP1-related intellectual disability, which is caused by a disruption in another gene, SYNGAP1, and has a similar phenotype of developmental delay, autism, epilepsy, and sleep disturbances. And we had 24 subjects with this diagnosis. And so we looked at the sensory profile of these patients, and what we found is that they were severely impacted. So what you're looking at here is the raw score on the short sensory profile 2 on the x-axis, and the number of patients, either Phelan-McDermott patients in black or SYNGAP1-related intellectual disability patients in blue on the y-axis. And the green box, I'm sorry, on my screen at least, the key is not coming through as well, but the green box is the score range of typically developing children. And so what you'll see is that both of our monogenic neurodevelopmental disorder populations were well outside the normal range, confirming that there is a high percentage of sensory impairment in these populations. But what was interesting to me is that we found that it was indeed less heterogeneous. So what you're looking at here are the specific categories of seeking and avoiding. And we'll talk more about the short sensory profile too in a bit. And you can see that almost all of these children scored in the much more or a little more than others, so to the right or up, in both categories, and that none of these children scored in the just like others category for both spectrums. And so we see the specific phenotype or specific pattern of sensory impairment in individuals with SYNGAP1-related intellectual disability. And I'm really excited that this paper was just published 2 weeks ago. You can see the reference on the bottom. It's in Brain Sciences. So if you're curious, I hope you'll take a chance and go read more about it. So now that we've shown you, or I've shown you, that there are specific patterns of sensory impairment, and hopefully I've convinced you that they are common and important, I'd like to give you the tools to screen for these sensory impairments in clinic. And the simplest way to do that is with parent questionnaires. The 3 that I recommend are the short sensory profile, the short sensory profile 2, and the sensory experiences questionnaire. So the short sensory profile looks like this. It's a 2-sided piece of paper with 38 questions. For each question, the parent answers on a 1 to 5 point Likert scale how frequently the behavior occurs. It's divided into 7 sections, each addressing a specific sensory feature or sensory modality, like we talked about earlier. And I really like the short sensory profile if you're trying to understand which sensory modality is most impacted. And that can be important for helping identify areas where parents can make changes to improve the child's quality of life. It takes about 10 minutes to administer, and you don't even have to be present. The short sensory profile 2 is similar. It's only 34 questions, similar 10-minute administration time, but the same format, a 1 to 5 point scale where the parent answers how frequently a specific behavior occurs. The big difference is that unlike the short sensory profile, the SSP2 is divided into behavior and sensory, and centered around these 4 quadrants, which come from the Dunn model of sensory processing. In this model, we think about seeking versus avoiding, which you can sort of think about as hyper versus hyposensitivity. versus registration versus sensitivity. Is your child not aware that things are going on around them, or are they ultra sensitive to even the slightest noise? And it's where you fall across these quadrants couldn't be very important for understanding the underlying circuit mechanism. Is it a gain control? Is it a registration or encoding problem? And I don't have a picture of the sensory experience questionnaire here, but I like it because it's a combination of the two. It kind of does a little bit of sensory modality and a little bit of hypersensitivity. I'm so sorry. We seem to have jumped ahead on my screens, but there are two other things you can do. One is direct sensory testing. And my favorite recommendation for that is you can take a cinnamon stick and put it in a glass jar and put that in your bag with all your other tools. It's a great way to test olfaction, which is the least commonly tested sensory modality in the clinic, but very, very important. And finally, you can just ask. So you can see I have some example text here, which is how I ask in the clinic. I'll let you read that yourself. But what I would want to share with you is that every time I ask this question, parents just come forth with so much information about how much these sensory issues impact their child's quality of life, and they are so appreciative that I've taken the time to raise this issue with them. So I hope you feel empowered to start screening for sensory impairments in your patients in your clinic. And I should say this goes for adult neurology patients as well as pediatric. So I hope you feel comfortable now with describing patterns of sensory impairments in various neurodevelopmental disorders, and that you feel empowered with the tools to identify these challenges in your patients. So I want to use my last few minutes to talk about how we use this on a research basis to look for identify targeted treatments by focusing on mechanism. So in our lab, we're most interested in autism spectrum disorder. And again, thank you, Dr. Sai, for setting this up so I can go pretty quickly. Autism spectrum disorder is diagnosed based on meeting criteria A, social and communication impairments, and criteria B, restricted repetitive patterns of behavior, which includes unusual sensory interests and reactions. And here is the text, specifically from the DSM, of what they mean by unusual sensory interests, which is hyper or hyporeactivity to sensory input or unusual interest in sensory aspects in the environment. And then they have some examples. So it is well documented in the literature, and I'm just going to show you a few examples here, that children with autism have deficits in a variety of sensory processing abilities. This is data from the short sensory profile. You can see each section on your left. And then these columns show what % of children in the study with either ASD or typically development had typical performance, probable difference, or definite difference. And what you can immediately see is that if you look all the way on the right in the definite difference column, there's most of the children with autism fall into that category, and very few of the typically developing children do. Similarly, when we look at this and here it is boxed in red. And when we look at this in a bar graph format, again, white is normal, gray is borderline, and black is severe. On the y-axis, we have the % of children, and the x-axis is the different sensory categories. We again see in children with autism, all those black bars are really high. They are impacted in many different areas. So we wanted to understand what are the, and here's the key. What are the distributions or patterns of sensory impairment in autism? Such a heterogeneous disorder, but are there specific patterns of sensory impairment? And to answer this question, we took advantage of the Autism Speaks Missing database, which for anyone out there who's interested in autism research, I strongly recommend. The Missing database has whole genome sequencing from over 11,000 individuals, representing more than 4,000 families. So it's both the affected individual and their parents and siblings. And we were excited because 378 of those children, or those participants, had provided short sensory profile data. And so we first started by just plotting it. What did it look like? And similar to previous reports, we saw that there was a broad heterogeneous distribution. So each of these boxes is one of the categories of the short sensory profile. The blue is the definitely different range, and the green is the typical performance range. And you can see that there are children, number of subjects on the y-axis, across the spectrum, definitely different or typically different. So then we looked at, for each individual child, what was were they typical? Were they different? What was their pattern? And what we found was that there 10 patterns of sensory differences described almost half of our cohort. So it was not random. It wasn't just that you had differences in some sensory areas and it was completely random. Most of the children fell into one of these 10 patterns. And in fact, there were only 87 patterns represented in our cohort of 378 children. So impairments of sensory processing are not random. They fall in specific patterns, suggesting that there are specific underlying circuit changes. We then did a cluster analysis, and in our cluster analysis, we've identified 6 distinct patterns of sensory impairment. So what you're looking at in this diagram, I'm actually showing you a sub-analysis where we looked at hyper and hyposensitivity in these 4 sensory modalities. Each bar graph or each set of bar graphs is a cluster. On the top left, labeled 1, is the cluster that was impaired in all hyper and hyposensitive in all sensory modalities. That's those 54 kids who were impaired in everything. Cluster 4 are children who have no impairments. They scored in the typically developing range in basically all categories. But then if you look at the other clusters, they're very striking. Cluster 3 is isolated hyposensitivity in the auditory spectrum. Cluster 5 is isolated hyposensitivity in hearing and touch. And so we were very excited to see these clusters because it suggested that there are specific patterns suggesting specific mechanisms. So then we looked at, in each of our clusters, what are the genetic variants that are present? Oh, I'm so sorry about this graph. But what I want you to focus on is that the subgroups are listed on the y-axis, or the x-axis, and we're looking at percent difference in variant frequency. And so the take-home message here is that there were specific genes that were selectively more likely to have a variant in each subgroup relative to the other subgroups, suggesting that there might be something about those genes that was causing that specific pattern. And I'm just going to show you one example here. This is subgroup 6, which, as you can see, is characterized by hyposensitivity in the auditory spectrum and hypersensitivity in taste. And on the right, I'm showing you the specific pattern of definite, probable, or typically different for the 7 sensory areas on the short sensory profile. And what we found is that in subgroup 6, patients were 30% more likely to have a variant in SHANK1 and 23% more likely to have a variant in SPAG7, suggesting that something about these genes might be related to this specific specific pattern of sensory impairment. And so, based on this idea that a gene and a sensory feature might be correlated or related, we have 3 different directions that we're moving towards. So the first is we want to take heterogeneous groups, like children with autism, and look at many different features, not just their sensory profile, but their age, their gender, their seizure burden, their sleep patterns, and ask, do patients within each of the sensory-based subgroups that we identify with our cluster analysis share other overlapping clinical features? And then secondly, we want to go the other way. Take groups of children with specific monogenic neurodevelopmental disorders where we can identify a specific, less heterogeneous pattern, and then do direct sensory testing to confirm that. And ask, do these patients with variants in identified genes exhibit the expected sensory patterns? And I'm super excited. We're already doing a lot of this. The study I just shared with you that we did with Dr. Holder and Phelan McDermott and SYNGAP1. We also have ongoing work looking at children with cerebral palsy in a collaboration with WashU. And then we also have just started looking at MECP2 duplication syndrome. And so finally, the thing that is nearest and dearest to my heart is taking this back to mouse models. So this is from a review that we recently did on olfaction in neurodevelopmental disorders. And it's really straightforward and simple to measure sensory processing, such as olfaction, in a mouse model. You can measure odor detection using a buried food assay, odor sensitivity using cross-habituation, and odor discrimination using a variety of tasks. And so here, we want to take the genetic variants that we identify in our clinical cohorts, put them into mouse models, and then see if we can replicate the sensory features that we see in the clinical population to understand if those correlated genes are causative of the sensory changes. So in summary, I hope I have convinced you that sensory features are common and debilitating in our patient population, that they are less heterogeneous in monogenic neurodevelopmental disorders, and that they occur in specific patterns which are associated with variants in specific diseases or specific genes. And for all these reasons, we believe that sensory features are an ideal biomarker for identifying clinically meaningful subgroups in neurodevelopmental disorders. I know I'm out of time, but I really would like to thank my lab. I know I didn't show you any mouse work today, but none of the work that I do would be possible without my mentor, Benjamin Rankiel, the whole Rankiel lab. My clinical and research mentors, as well as my funding sources, are shown here. And I would be delighted to answer any questions.
Thank you, Dr. Lyons-Warren. Wow, we are learning so much this morning about topics that I think are completely underappreciated, like sensory challenges. So we have some great questions from the audience. One is, could this be a signal-to-noise issue? Is this, that a person can't distinguish an important sensory input from other environmental inputs? And then a second part to that question is about involving different sensory modalities, sound, touch, position.
Yeah, I think that's a great question. And that's actually exactly why I think it's so important that we do a better job of characterizing which sensory modalities are involved and whether it is hyper or hyposensitivity. Because you could imagine that there's a child who's hypersensitive to everything, to sound, to touch, to light, and that's probably a gain control issue, or perhaps, like you're saying, a signal to noise issue. But if there's a child who is both hyper and hyposensitive exclusively in the auditory domain, that suggests to me something more in the auditory cortex, or maybe the the thalamus, where there's processing specifically of auditory information occurring. And so understanding the specific phenotype can give us clues to where we need to look for the circuit disruption. This signal to noise question, my graduate work was actually sort of related to the cocktail party problem, which even those of us who hopefully don't have neurodevelopmental disorders often experience, where you can't, you're in a cocktail, like none of us do this anymore, but a large party with many people talking, and you can't focus on the person who's right in front of you because you can't separate that background noise from the relevant stimulus. And we don't really understand the circuit mechanisms that underlie that, but it's probably very similar to the much more debilitating sensory disabilities that our patients are experiencing.
Excellent. Another question is there evidence of quantifiable sensory deficits in Rett syndrome? And could sensory phenotypes be outcome measures for clinical trials?
I promise this is not a plant question, but it is something I talk to Dr. Suter and Dr. Pelivan about all the time. So yes and no. There are documented changes of olfactory epithelium differences in mouse models of Rett syndrome. The problem with measuring sensory impairment in patients with Rett is that they're actually very impaired. And it's difficult for us to measure sensory impairments using a parent feedback survey in individuals who have a developmental age less than 6 months. There really are no good validated tools below that age. And so we're doing this in MECP2 duplication syndrome, where the children are a little bit less impaired and there's more variability, to see how can we characterize sensory features in those patients, and then hopefully that will be extendable to the full Rett population. The other thing is, yes, I firmly believe that sensory features are an ideal biomarker for use during clinical trials because they are directly related to clinical function, how well a child does in real life, and they change over time. So we can measure them serially and see that they're improving or getting worse or potentially correlating with other functional outcomes. And they're easy to do. It's a 10-minute survey in the clinic. I just got to put a plug in here. We're actually in the process of developing a new sensory survey, which will specifically look at hyper and hyposensitivity in a sensory modality specific manner. And our goal is to be the first sensory survey that is validated against direct sensory testing, which has not previously been done. So keep an eye out for that. And if you have kids you want to participate, send them my way.
Absolutely. Yeah, that's, I will be sending all my kids to you. I had a question about how our current therapies for sensory challenges, do those create long-lasting changes in the brain, or are they sort of just symptomatic treatments at the time? Thinking about occupational therapy type methods.
Yeah, that's a great question. I don't know the answer because no one's looked at how the child's function in terms of sensory impairment correlates with like magnetic MRI imaging or functional MRI imaging. So I don't know if it's creating permanent changes in the brain. We don't really see that like fixing sensory impairments fixes other functional outcomes. So if I improve your sensory sensitivities, your language doesn't get better. It improves your quality of life, but it's mostly training right now. So like habituation, you have difficulty with loud noises. We're going to expose you over and over and over again to loud noises until you hopefully become less sensitive to them. Or child is bothered by light. So we're going to make an environmental change and put heavy dark curtains in your bedroom so you can sleep better. So I think we don't have good treatments yet. And I hope my long-term goal is that we can figure out the underlying circuit mechanism that's impacting the sensory features, which are full kudos to everyone else who spoke today. I think it's easier to study sensory processing because it's so easy to do input and output. And then once we have that circuit mechanism and we're treating the circuit problem, then that will also impact the other circuit problems that are causing the other features.
For the adult neurologists in the room, I'm curious about if there are disorders in adult neurology where people develop sensory hyper or hyposensitivities. Of course, COVID might be one of those with the loss of taste and smell. But I'm curious about other, what people might be looking for in adult patients.
Yeah, so I think the one that I always remember as a med student is that loss of smell is one of the earliest signs of Parkinson's disease. But the other thing is that oftentimes these sensory impairments are present in childhood and may go unrecognized. And then later on, an adult would develop a more fulminant form that would then have sensory features associated with it. And in retrospect, they'll say, oh yeah, I was always like super sensitive to touch or something. In our pediatric population, the children sometimes outgrow. So I have parents tell me, oh yeah, my kid used to be very sensitive to sound, and now he seems to be less bothered by it, or she. And I think part of that is that we make adjustments. And so it's less obvious to the parent. And again, without direct sensory testing, without measuring the sensory sensitivity or sensory threshold, it's a little bit hard to know.
Can you comment on the transdiagnostic aspects of measuring sensory symptoms? I typically think of, if I have a child who doesn't have any sensory symptoms, I think, well, that kid's probably not autistic. But that's not necessarily true, and the opposite can also be true. So I'm curious your thoughts on that.
Yeah, I think, are you asking me about the use of sensory features as a diagnostic feature in determining the specific diagnosis?
Yeah, I guess there's two parts to my question. One is, how much does it help contribute to specific DSM-5 diagnoses? And then I guess the other question is, how much do these overlap? And can you tell the difference between sensory sensitivities from Tourette syndrome versus autism?
Yeah, so that's a great question. Currently, no. One of the things that I try and do is to show people that there is literature out there that, like, one study where they showed this was the most common sensory modality, but it is not part of DSM criteria as of yet. And I think it's just that we haven't done enough detailed work. We mostly say present or absent sensory impairment and leave it at that. But what our work has shown is that there are specific patterns, and I do think it could be helpful. And the truth of the matter is you're going to get genetic testing, right? Like, that's the world that we live in, and that's amazing and great for us and for our families. But at the beginning, when you're trying to narrow your differential, I do think asking those specific sensory questions can help point you, oh, this is more like Phelan-McDermott versus SYNGAP1 versus hopefully MACP2 duplication. You know, you're going to use the movements as the primary diagnostic criteria in Tourette. But when you're trying to distinguish, does this child have a comorbidity of autism, or do they just have auditory features that we know are already associated with Tourette syndrome? That's where I think it can be helpful in helping you determine how many diagnoses are present.
We're over time, but I have one last question about just practical nature of how do you bill for sensory processing disorder when it doesn't exist as a diagnostic code?
My chair, Dr. Clark, is listening, so I probably shouldn't say this, but I don't. My primary concern is the patient, and I'm fortunate to work in an academic center where I have the luxury of time to ask these questions. We need to push, right? That's what Texas Neurological Society can help us do. We need it to be recognized and billable, because if you're going to spend time on it, you should get credit for that. And it's an important thing that you should be spending time on.
Amen. All right. Well, thank you so much, Dr. Lyons-Warren. Really appreciate it.